Good morning. My name is Lanxin Zhu from Professor Peng's group. I'm very happy to be invited by AI Microscopy Symposium to give today's talk on high-speed 3D imaging and analysis of biological dynamics using AI-based light field microscopy. Our research basically aims to address the current challenge in capturing transient biological dynamics, such as neural activities and heartbeat in 3 dimensions. Here, I will introduce how our deep learning-enabled light field microscopy can break the spatial-temporal resolution limit, realizing fast and accurate 3D imaging. Then, we will demonstrate its applications in recording fast biological dynamic processes occurring in live model organisms. I will first introduce the remaining challenges in current 3D microscopy implementations when doing live imaging, and how we are motivated to develop this AI-based light field microscopy approach. As we all know, many cellular processes occur in millisecond timescale. For example, the blood flow in the beating zebrafish heart, the neural activities in freely moving C. elegans. High-speed and high-resolution observation of these dynamical biological processes in 3 dimensions are very attractive to a broad range of life science researchers in neuroscience, cardiology, and developmental biology, as many mechanisms hidden behind the phenomena could be possibly revealed through using advanced imaging. In other words, it will be extremely helpful if you can in total capture the transient signals with high quality. However, current 3D fluorescence microscopes, either epifluorescence microscopy such as confocal and two-photon, or light sheet microscopy, require mechanical scanning to record a number of 2-dimensional images. This scanning-based microscopy suffers from limited imaging speed and substantial photobleaching. The achievable temporal resolution of these 3D microscopes is seconds to minutes timescale, which is insufficient to capture the millisecond transient processes. It's highly valuable if we have a scanning-free 3D microscopy with high volumetric speed. So I will introduce light field microscopy, which is an emerging high-speed 3D imaging tool. And what is light field microscopy and how it works? As we all know, if you look at the ordinary wide field microscope, you can obtain a clear image only when the signals are in focus. This is because for each point on the sample, the light with different angles will eventually merge together, and the angular information is indistinguishable. But for light field microscopy, by inserting wavefront sensing elements at the native image plane, such as macrolens array, the light with different angles will be reallocated onto different pixels of the camera sensor. Then, different pixels behind each lenslet will represent different angles, while the pixels behind each different lenslet will represent the lateral position. So the 2D image plane now becomes a map with both position and angular information recorded. This light field image, encoded with both lateral and angular information, enables us to extract the 3D structures by the post-processing algorithm. That's why light field microscopy can realize volumetric imaging by only one snapshot. In a word, light field microscopy is a novel volumetric imaging method that basically includes two stages. At first, capturing the light field image, and secondly, computationally reconstructing three structures from it. So how to acquire light field image? It can be easily achieved by inserting a microlens array or other sensing elements at the native plane of an ordinary wide field microscope. With eliminating the need of extra scanning, the volume rate can be increased by 2 to 3 orders of magnitude. This advance enables the observation of the millisecond transient dynamics. Since light field microscopy can provide high-speed volumetric imaging, limited only by the camera frame rate, it has delivered promising results in various applications, including the recording of neural activities in C. elegans mouse brain and visualization of cardiac dynamics in model organisms. However, the image quality prevents its wider applications for capturing millisecond processes at single-cell resolution. Current light field reconstruction merely follows the light field deconvolution, which models the formation of light field image using a wave optic model and adopts the iterative deconvolution scheme to infer 3D images from 2D light field raw data. One problem of this method is the low and non-uniform spatial resolution in the results and the presence of reconstruction artifacts. As the figure shown, the quality of the images at different depths is not uniform, and the red frame indicates the artifacts near the focal plane. This is caused by the non-uniform distribution of the light field along the depth. As the high-frequency components of the light field are too sparse at some depth to infer the angular information, the resolution of the reconstructed image will be degraded, especially on the focal plane. The second problem is the large computation cost and the low reconstruction throughput. Since the method is based on iteration, the reconstruction process can be very time-consuming. Sometimes the dynamic raw data recorded in few seconds will require a few hours to be processed, which compromises its potential for long-term applications. While these problems can be mitigated by optimizing the hardware or software, such as moving the position of the microlens array, recording dual-view light field at an orthogonal angle, and separating the sparse components of the light field, the extra system complexity could be the wide dissemination of the light field microscopy. So here in our work, we focus on developing a new reconstruction algorithm to break through the bottleneck of the light field imaging, which can easily be applied on a basic light field microscope and utilized by common microbiological labs. Here we are going to show how we break this limit of the current algorithm. Following the success of deep learning in biomedical imaging field, it's natural to think that, can we design an artificial neural network to do a better job on this? We developed the following pipeline. First, We use other scanning-based microscopes, such as confocal microscope, to acquire high-resolution images of static samples. For example, an anesthetic worm. Using the wave optics model of light field microscopy, we projected these 3D images into synthetic light field images, which were then used as input for network training. By iteratively minimizing the loss of spatial resolution by incorporating abundant structural information from training data, the network could be gradually strengthened until it's capable of deducing 3D high-fidelity signals at uniform resolution across the imaging depth. Once the network is properly trained, it can deduce 3D volume video from a sequence of light field measurements at a millisecond temporal resolution. Meanwhile, the network can directly deduce 3D text at video rate throughput. Therefore, it's suitable for time-lapse video processing. If we want to build a network to reconstruct 3D stacks from a 2D image, it's important to extract the angular information of the light field image and then efficiently transform them to the depth images. For each light field image, which contains both lateral and angular information, we interpret it as a collection of views. By looking at the light field, we look at the objects with different views of different angles. This figure illustrates this connection. By taking pixels with a certain location regarding each lenslet, for example, we take all the pixels labeling red. We formed a low-resolution view of the volume at a certain angle, and we then need to transform these low-resolution view images to high-resolution depth images by deep learning. We built a view channel depth network termed VCDNet based on a UNet model. The network can transform the views into depth images. We first set a group of sub-pixel convolutional layers to extract the views from the light field images, and interpolate them to pair with the depth images. Then the features were extracted and incorporated into multiple channels in each convolutional layer, and the final output channel were assigned to a number of planes representing different depths to generate an image stack. It's noteworthy that this is the first time in the world to directly transform a 2D light field image into 3D stacks by deep learning. To validate the performance of our network and demonstrate its biological applications, we built a customized imaging system based on an upright wide field microscope. It enables light field and wide field dual-channel imaging by using a beam splitter, so that we can test the accuracy of our reconstruction. For light field imaging, we added a microlens array as the native image plane to collect the light field signals. Our customized system can also realize dual-channel imaging. Combining our customized light field imaging system with our VCDNet, we can validate the performance of our method. One advantage of deep learning method is that since the network is training data driven, the resolution of reconstructed results is not limited by the non-uniform light field and diffraction limits. Once the network is properly trained with the high-resolution label data, the resolution of the reconstructed images can approach the label data as close as possible. Therefore, the algorithm is fundamentally not restricted by diffraction limits of the optical light field system. We tested the spatial resolution of our method and compared with the light field deconvolution. We reconstructed the fluorescent space captured by our customized light field microscope and measured the point spread function as our spatial resolution. As the left figure shows, the space reconstructed by deconvolution algorithm show artifacts on the focal plane, and the blue lines in the right figure indicates its non-uniform axial resolution. On the contrary, our VCD shows artifact-free and uniform image quality, achieving an isotropic resolution of around 1 micron, which is similar to the high-resolution label data. Our VCD also shows superior performance on the synthetic tubulins. As the figure shown, while the light field deconvolution shows low axial resolution and some artifacts, our VCD net is capable of reconstructing the tubulins with nearly isotropic resolution. In addition to the greatly improved image quality, our VCD net also shows significantly higher reconstruction throughput. After training on gigavoxels of data, the well-trained network was capable of inferring a sequence of 3D volumes at a rate up to 13 volumes per second, without the need for any iterations or hyperparameter tuning. The reconstruction throughput is validated to be 3 orders higher than the deconvolution algorithm, achieving real-time reconstruction. For example, 1 minute's recording of freely moving C. elegans will yield 6,000 light field images at the volume rate of 100. With our network, it only takes around 7 minutes to reconstruct, while the deconvolution algorithm needs more than 100 hours, almost 4 days. The high reconstruction throughput of our method is exclusively suited for observing long-term processes. In addition to the validation on simple samples or synthetic data, how about the performance on biological samples? As I mentioned at the beginning, the observation of neural activities and blood flow is crucial for the research in neuroscience and hemodynamics. C. elegans are used extensively as the model animal in neurosciences. It could be a good demonstration of the imaging speed if we can capture the whole body of an unconstrained C. elegans. To validate the accuracy of the reconstructed signals, we first tested the pipeline on the immobilized worms. As the upper figure shown, high-resolution neuron images can be accurately reconstructed without artifacts, and the neurons can be better identified by the deconvolution method. Intensity accuracy is also crucial for calcium imaging. We applied our method on synthetic neurons with time-varying 3D positions and intensities, reflecting calcium signals. With referring to the ground truth, our VCD results show high intensity accuracy up to 99%. The capability of our VCD net enables us to demonstrate the calcium imaging of freely moving C. elegans for a long timescale. The neuron calcium signaling of the fast-moving C. elegans can be recorded at an acquisition rate up to 100 Hz, yielding 6,000 light-field images in both GFP and RFP channels in a 1-minute observation. As the figure shown, the neurons reconstructed by the deconvolution method cannot be distinguished, while each neuron of our method can be clearly resolved. The video also shows the capability of the VCD net to reconstruct the neural signals at single-cell resolution during fast body movements. Based on these videos with single-cell resolution and long timescale, we successfully extracted the 4D spatial temporal patterns of calcium signaling and calculated the worm's velocity and curvatures related to its locomotion and behavior. The upper right plots show the calcium fluctuation of different neurons along the worm body. We found the patterns of calcium signaling to be relevant to velocity and curvatures, shown in the lower right. This calcium analysis in 3 dimensions needs 100 Hz volumetric imaging speed and a single-cell resolution, which is unachievable for most of the current 3D scanning microscopes. High-speed and high-resolution observation of the beating zebrafish heart is also a great challenge for current 3D microscopy. So we applied our method to capture the beating zebrafish heart. To reduce the background from body tissue, we generated a rod-like laser beam to selectively illuminate the heart region. We recorded the light field video of red blood cells and beating cardiomyocytes at a volume rate up to 200 Hz. And the light field video was then successfully reconstructed at single-cell resolution with our VCD net. This VCD reconstruction with single-cell resolution enables us to observe the flow pattern of the blood cells with the heartbeat, which is unmatched by most of scanning 3D imaging methods, and thus permitted quantitative investigation of transient cardiac hemodynamics. While deconvolution method suffered from suboptimal cellular resolution and artifacts around the focal plane, as shown in the left figure, the blood cells reconstructed by our method can be clearly distinguished even in the regions where blood cells are densely distributed. This VCD results with single-cell resolution enables the tracking of each red blood cell through the cardiac cycle. We track the red blood cells throughout the entire cardiac cycle of around 400 milliseconds, during which the blood was pumped in and out the ventricle at a speed of over 3,000 microns per second. The trajectories are shown here, with the color representing different velocities. Each arrows here show the direction of the blood cells' movements. In addition, we also analyzed the ejection fraction of the heartbeat, with segmenting the continuous heart boundary as the dotted lines in the upper right image shown. We also successfully quantified the volume change of myocardium and ventricle during the diastole and systole. As the lower figure shows, we mapped 4 heartbeat cycles and calculated the ejection fraction of the heartbeat to be around 71%, which is the volume change ratio of the ventricle during the diastole and systole in one cardiac cycle. We also imaged the blood flow of zebrafish tail vessels and analyzed the hemodynamics. The flow pattern of the red blood cells through the intersegmental vessels can be clearly visualized in the left video, and we automatically tracked the trajectories of flowing blood cells. The red blood cells are denoted by red bars, and the colors on the trajectories represent the maximum traveling speed at that position. In summary, our AI-based light field imaging method, termed VCD-NET, can break the spatial-temporal resolution limit, realizing fast and accurate 3D imaging. It's capable of capturing 3D long-term transient biological dynamics with single-cell resolution and reconstructing at video rate. These advances are crucial for sustained volumetric imaging of transient processes, such as neural activities of freely moving C. elegans and hemodynamics of embryonic zebrafish. As acknowledgement, thanks to Professor Fei Peng and my colleagues' work, our lab is also doing research on light sheet microscopy and microfluidics. Feel free to contact us if you have questions. We also appreciate our important cooperators, Professor Zong Sai, for providing zebrafish sample, and Gao Shangbang for providing the C. elegans in our research. That's the end of my part. More details, please see our latest article on Nature Methods. Thanks for listening.
Wonderful. Thank you so much, Lanxin. Appreciate very much the effort you put into preparing the presentation, and amazing work, so high impact. This kind of problem of imaging large and freely moving either samples or organisms or smaller samples is really tricky. I remember myself trying to image blood vessels in living mice using intravital microscopy, and that was extremely challenging, to the point where this is now 15 years ago, we had to essentially do it by hand. We couldn't even count on algorithms because the jitter in the sample was so high. We basically had to go with digital tape, frame by frame, and count how many frames and how, how far each red blood cell was moving to get blood flow measurements. With this kind of technology, you can certainly do much, much better. So there are a couple of questions that came in. One from Zhou Yao, and the question is about how generally applicable this solution is. And this is this question comes up a lot when talking about machine learning and deep learning applications. But that's the question here. How generalizable is the model trained on the sample and applied to another? Can a model be trained on many data sets and generalize well? So it would be interesting to hear what your ideas are there and what your experience. Perhaps you can give us a sense of how many images you've used for training. That kind of thing would be helpful.
Hello, I'm Zhu Lanxin. Thanks for the professional questions. We tested the generalizability by demonstrating the hybrid data and cross-sample training of like red blood cells, myocyte nuclei, and myocardium. We also demonstrated that our VCD could generalize well when trained on many data sets and applied to many samples. For example, we trained a hybrid network containing the training data of red blood cells, myocyte nuclei, and myocardium.
Mm-hmm.
It can reconstruct 3 types of data with good image quality. We also demonstrated on the cross-sample applications, like we reconstruct the nuclei by the network trained on red blood cells. The result shows that when the types of signals are similar, like the red blood cells and nuclei, which are both point-like signals, our VCD net are doing well. And when the signal types are completely different, like the red blood cell and the myocardium, our model is also highly generalizable after being supplemented with the transfer learning based on only small amount of target data involved. More details, please see the supplementary note in our Nature Method paper. Thanks.
Yeah. That paper is really very nice. There's a lot of videos. I encourage you, and you can also get the data set, at least one of those data sets I have played with, the one with the heart, where you see the heart beating. That's a pretty impressive data set. And it, if there's, and I know there are people here that do this kind of imaging. When you look at it, you will be surprised to see there is nearly no jitter. You almost kind of think, how is this possible that the heart is just still there, and you can see all the detailed movements of the heart without the typical, the animal movement itself. With that, I had a question, or I'd like you to comment on the availability of the code so that others can try this out. Perhaps you can elaborate on that, the code and perhaps training data or testing data.
We have updated our code and training data on the GitHub, and the link is available on our Nature Method paper.